Want to go ahead and start recording. Welcome everyone to the third Marine Science Seminar, whether you're here today or I guess watching the recording of this at some later date. Welcome. On behalf of FIU, I'd like to welcome Dr. Jennifer Sneed to talk to us today. She is a research scientist at the Smithsonian, as you can see there on the title slide. She did a MSc at USF in Tampa, and then a PhD in Germany, and is now in Fort Pierce, looking at using a whole range of techniques, molecular biology and bioinformatics, to look at ecological interactions in the marine environment. And today she's going to talk to us about a topic close to my heart, which is the future of coral reefs. So without further ado, I'll hand over. Thanks again, Jennifer, and folks can put questions as we go in the chat, and I'll maybe moderate those at the end. Otherwise, people can unmute themselves and ask questions. So thanks again, and take it away.
Okay, thank you for the introduction. I'm gonna minimize some video screen so I can see my talk. So thank you, Alistair. As you said, my name is Jennifer Sneed. I'm from the Smithsonian Marine Station in Fort Pierce, Florida. So I'm not sure how many of you know, but we are a field station for the National Museum of Natural History in DC. So we're actually a small field station located in Fort Pierce. There's about, I would say, about 10 permanent staff, 10 to 15. And so it's pretty small. And then we have postdocs and interns and things like that on occasion. We are located in Fort Pierce, which is just a couple hours north of Miami, for those of you who don't know. And I'm sure if anyone would like to ever come up for a tour, just get a hold of me and we could probably arrange something. So today I thought I would talk about some of the work that I started at the Smithsonian Marine Station back in 2011. So it's been a little bit now. I started some of this work right after my PhD, trying to understand some of the ways that bacteria are involved in mediating the interactions between benthic marine organisms. And so most of my work at the Smithsonian has been on coral reefs. I did a lot of work before that looking at green algae and some of the microbial communities associated with green algae. But today I want to talk about some of the work that I've been doing looking at coral larval settlement and the role of bacteria in that process. Here we go. Okay, sorry. So I just wanted to start out with a really general sort of overview of these sort of interactions that I'm interested in. And so as a big umbrella topic, it's sort of bacteria as mediators of benthic interactions. And so a lot of what I did for my PhD work involved looking at the host organism and the chemicals, the secondary metabolites, natural products that it was producing that impacted the associated bacterial community. And so, here we go, yeah. So organisms can produce nutrients or chemical attractants or a number of different things that can attract certain bacteria to associate with them. And I think it's pretty well established now that nearly every organism has some sort of microbiome associated with it, either within its cells, on its surface, but in some way associated that's very important. And we're learning a lot about that, especially now with the human microbiome. We know that we actually have more bacterial cells in our bodies than we have human cells. So the microbiome of an organism is critically important, and we're learning more and more about that. And so that organism, whatever the host organism is, whether it's a human or a seaweed or whatever, it can produce some sorts of compounds that'll attract certain microbes to its surface or to its organism. But it can also produce compounds that are deterrent. And so things like antibiotics that are going to specifically deter certain bacteria from getting into the organism, whether those be pathogenic bacteria that it wants to sort of mediate, or other sorts of bacteria. But one other area that I'm really interested in is what those bacteria are doing for the organism once they do have their established microbiome. So once that organism A has its microbiome associated with it, those bacteria themselves produce natural products that affect the interactions in the environment. And so those organisms, those bacteria can produce antibiotic compounds. And an example of this from the literature is the elkhorn coral here, Acropora palmata. It has a bacterium associated with it called Marinobacter that produces antibiotic compounds that actually help fight off this disease, this white pox disease, Serratia marcescens. We're also in our lab working now, I've been working a lot with Blake Ushijima, who's working on trying to identify probiotic bacteria, so bacteria isolated from healthy corals that can fend off the stony coral tissue loss disease that's been so rampant along the Florida coast and now into the Caribbean. So we're working on some more of those sorts of interactions in the lab now as well. I find this really interesting, and I'm not sure how many people know these sorts of things, but there are bacteria associated with different organisms that produce compounds that can actually deter feeding, so they can protect the host organism by producing compounds that keep the host organism from being eaten. And so this is, again, just an example from the literature, but this bryozoan, Bugula naratina, produces these compounds called bryostatins, and the bryostatins actually act as feeding deterrents for the bryozoan. And it was thought for a long time that it was the bryozoan itself that was producing the compounds, but then it was later discovered that it's a bacterium living inside the bryozoan, this Candidatus endobugula certula, that's responsible for producing those compounds that act as a feeding deterrent. And these studies were done with fish. So this example involves the French filefish was the model predator in this study. The bacteria associated with the organism can produce compounds that inhibit settlement, so keep something from settling on its surface. And one of the examples from literature of this is the sea lettuce, Ulva lactuca, has a bacterium associated with it called Pseudoalteromonas tunicata. And in this study, that was shown to inhibit settlement of the larvae of that bryozoan, Bugula naratina. And then the final example I want to give are compounds that are produced by the bacteria that actually induce settlement. And so that's what I've been working on for the last 9 years. Is looking at these compounds that are associated with the surface of crustose coralline algae that induce settlement of coral larvae. And so you can see this little swimming larva over here. It's in a petri dish, so it keeps hitting the sides of the petri dish. But that's what it looks like under magnification. And so that's what I'm going to spend the rest of the time talking about, is some of the work that I've been doing looking at those factors. So the bacteria associated with crustose coralline algae that influence settlement of coral larvae. And so just to take a big step back to why do we care about the bacteria that are involved in helping to induce coral settlement? Well, I suspect that all of you know the coral reefs are not doing so great right now. I mean, we're in the middle of another bleaching event at the Great Barrier Reef. Seawater temperatures are kind of devastating the reefs on top of all the other things they're trying to deal with at the moment. And this is just sort of a really depressing quote from the Nature Conservancy that says 70% of the world's coral reefs will be destroyed by the year 2050. So obviously there's a lot that has to be done to mitigate some of those stressors and relieve the coral reefs from the stressors that they're under. But once that happens, we have all these degraded reefs that look like this, right, with nothing living on it left. And how do we get back to this healthy, thriving reef that you see here on the right? And one of the critical events that has to happen for that return to a healthy reef system is recruitment of new individuals to the reef. And so that's why we're really interested in this process of how coral larvae recruit, how they recruit to reefs, what factors are affecting that. So for those of you who may not know, corals reproduce in two different ways. Some of them are what we call broadcast spawners. Broadcast spawners release gametes into the water column. They have external fertilization in the water column. And then that new zygote develops in the water column into a planula swimming larva. Brooders, on the other hand, they have internal fertilization. So they actually either take in the sperm themselves or are parthenogenetic. And then they will release fully developed larvae already. And those larvae are competent to settle upon release usually. And so at that planula larva stage, then those larvae will swim down to the bottom and they will attach. And with any luck, they will grow into juvenile corals and then hopefully eventually to adult corals that will then reproduce again. But we're really interested in this stage going from planula larva to spat to this newly settled coral because it's such a critical stage for the survival of the coral. The choice of location for that settlement process can be critical to the survival of the coral to ever grow and reach maturation. And so here again, you can just see this is what I'm talking about when I'm talking about coral larval settlement. So you see the swimming larva on the left. It's going along the edge of the petri dish there. This is under magnification. And then it will attach to a surface and then metamorphose to this pretty little flower shape that you see on the right. And then that's the newly settled coral. We take the term spat sort of from other systems, but that newly settled coral will then grow to a, hopefully to a juvenile coral and then to an adult. So we know that coral larvae will respond to a number of different factors. So they will settle, some will settle on just biofilmed surfaces under the water. So often coral rubble, just that is biofilmed and has been sitting underwater. There's some evidence that some coral larvae will respond or settle in response to conspecifics, the presence of conspecifics. And there's a lot of evidence that they will respond and settle on crustose coral and algae, CCA. Just for the rest of the talk, I will probably interchange the terms crustose coral and algae and CCA. So if I say CCA, I'm referring to crustose coral and algae. So some of the stimuli that they actually use to identify these settlement substrates include color. So there's some evidence that corals can actually detect the color red and are attracted to the color red. They use light as a signal. So when it's time for them to move out of the water column towards the bottom, they will swim away from the light down towards the bottom. There's also some evidence that they can be attracted to reef sounds. Once they're on the bottom and they're sort of crawling around, they can distinguish textures, and then they can also respond to chemical cues. And so what we're really interested in our lab are chemical cues, mainly that are associated with crustose coral and algae. So this was some work done before I came to the lab. This was done by Raphael Ritson-Williams and others at the lab this was published in 2010. They were looking at the system and looking at the different CCA and how coral larvae respond to different species of CCA. And it turns out that while there's this huge, vast amount of evidence that coral larvae will often settle on CCA, it turns out they will show preferences for some CCA over others. So not all CCA are created equal. And in this study that Raf did, he looked at these 3 species of CCA, Hydrolithon borgesonii, Titanoderma prototypum, and Periogonolithon solubili. And it turns out that the larvae of the elkhorn coral, Acropora palmata, prefer to settle on Hydrolithon borgesonii and Titanoderma prototypum over Periogonolithon solubili. So they can actually make choices between the different species of CCA. So when I joined the lab then in 2011, I came from a real microbial background. So I was used to looking at the roles of microbes in these sort of chemically mediated interactions. And so from that perspective, I came to this project thinking, well, how are the larvae distinguishing between the different species of CCA? And based on my PhD work looking at green algae, I knew that typically different species of algae host unique microbial communities on their surfaces. And so I was interested to see if maybe these different species of CCA actually had unique microbial communities on their surfaces that may be acting as a way for the bacteria to recognize those different CCA from each other. So that was sort of the first thing I did when I got to the lab, was to try to answer that question. And so I did 16S sequencing of the same three CCA species that Raf had previously worked on, and looked at the microbiomes associated with those different CCA surfaces. So what you're seeing here are just pie charts showing the different bacterial orders that were associated with the different CCA. It turned out that the different CCA did have significantly different bacterial communities on their surfaces. So these are all significantly different from each other. A couple of things that just jumped out at me from this data are the actinomycetes. So you can see here for Hydrolithon borgesonii, there's a really high amount of actinomycetes, and a little bit less there for the other 2 species. And the reason that really jumps out to me is that actinomycetes are known to produce a lot of interesting chemical compounds that have activity in the environment. So that's one of the things that sort of jumps out from this data set. And another thing that really jumped out at me is down here on this P. solubili, we saw a higher abundance of oscillatoriales compared to the other 2 CCA species. And this oscillatoriales is a group of cyanobacteria. And everything that I sequenced were microscopic organisms. There was nothing that you could see on the surface, but there are macroscopic members of this order that are known to inhibit coral larval settlement. So I think it's really interesting that on this CCA species that is less preferred by coral larvae, there is an increased abundance of some of these CCA that may be related to ones we know inhibit larval settlement. So then to sort of jump off from that point, so I could say, yes, these CCA have unique microbial communities. There are definitely differences in the bacteria that are associating with them. Could that be influencing the larval settlement? And so to do that, I had to jump to sort of a culture-based approach. So looking more at culturable bacteria as opposed to the unculturable bacteria that you're looking at through sequencing. And we had to do some settlement assays, obviously, with coral larvae. And so I'm going to walk you through how we actually get the larvae that we want to use for these assays. It's not trivial, but it can be fun, and it's definitely interesting. So for all the spawning species that we work on, we do all that work in Belize. So here you can see we have a field station called Caribou Key, and this is about 15 miles off the coast of Belize. This is a lab that belongs to the Smithsonian. It's pretty small, so that whole island is less than an acre large, and it hosts about 6 scientists at a time. Sometimes we try to squeeze a few more in during spawning, but typically there are only supposed to be 6 scientists there at any given time. And then there's always a cook and there's a station manager. So typically you have about 8 people on this island at a given time. And so in order to collect, to get larvae to use for experiments, we actually have to go out and collect the gametes from the water column. And so with these guys, we go out at night when they spawn. So these spawning corals that we work with typically spawn after sunset. So it's always at night in the dark. And they typically only spawn once a year. So most of the species we work on only spawn in August. Some are in September. And they typically will spawn 1 or 2 nights. So they might spawn 1 or 2 nights in August, and then they're done for the year. So it's a bit tricky. You have one shot at it each year for any given species, and then hopefully you get what you need to be able to do some work with it. So we actually go out every night within a window. So for example, for the Acropora species, we're usually looking 2 to 7 days after the full moon in August. So we'll go out in the water every night at a certain time, and we will start looking at the corals to see if we think they're going to release gametes. And you can see this is my colleague Zach Foltz here. And he's looking at this Acropora cervicornis to see if it's going to spawn. And the way that you know if it's going to spawn is that they will, the sperm and eggs for these are all compacted into what's called a gamete bundle. So it's this little pink ball, and you can actually see some corals have spawned already in this picture. So it's these little pink balls that are floating in the surface, or floating in the water. But when they're getting ready to spawn, they will put those gamete bundles in the mouth of the coral polyp. And so you can start to see these little pink bumps along the surface of the coral. And when you start to see those pink bumps on the surface, you know that coral is going to spawn, because you can see them coming up here. And then we'll go actually put a net over top of that coral and wait for the gametes to be released. And there you can see this coral has just released all its gamete bundles. Those gametes then those bundles will float up to the surface, and they will break apart at the surface, releasing the sperm and eggs, and then fertilization will just happen at the surface in the water. So here you can see this is a picture of the sort of nets we put over top of the corals when they're getting ready to spawn. And at the end of these nets, there is a container of some sort that the gamete bundles all float. So they will go up to the top of the net. They'll get collected in that container. Then we can remove the container. We take that into the lab, and then we do all the cross fertilizations in the lab. So we'll take eggs from one colony and sperm from another colony and cross fertilize. And you can see here, this is Dr. Nicole Fogarty. I've done a lot of work with her down at Caribou over the years, and she's doing some cross fertilizations here. So, and then this is our setup in the wet lab there at Caribou. So then we have these developing larvae in these buckets, and we sort of have to baby them for a few days while they grow. And eventually, after about 3 or 4 days, they'll grow into fully competent larvae that are ready to settle. And then we can actually use them in our experiments. So that was sort of a long explanation of how it is that we get the larvae from the spawners that we need to get to use in experiments. And it's not trivial. It's not super easy. So we tend to like to do most of our work with brooding species because they're much easier, and then sort of do all the double checks then with the spawners once we've got to some of our answers. So getting brooders is a lot easier. We do this work in Florida. So we work with this the coral Porites astreoides down at Mote Marine Lab in Summerlin Key. And for these guys, it's a lot easier because we can go out and we can actually bring these guys into the lab. We collect them out off the reef, we bring them in, and then when we're finished, we just take them out and cement them back down, and they seem to do fine. It doesn't really harm them. So we bring them into the lab, we put them into these mixing bowls in this setup here. You can see, so each mixing bowl has a water source that's flowing into it. The water will flow over the coral, and these corals will release larvae throughout the night. So they just kind of dribble the larvae out. And as they're releasing the larvae, then those larvae will flow over the handle of the mixing bowl. We've made them a little water slide. They will flow over the handle and into this container. This is a tripore beaker with Nitex mesh on the bottom. So the larvae get trapped in there, and then the water is continuously flowing over that. And so what's nice about this is we can collect the corals, we bring them in, we set them up for the evening. We do this, before we go to dinner, or maybe sometimes after we get back from dinner. We go to bed, we get up in the morning, and we have a container like this full of larvae. And the other thing that's nice about this is that these larvae are already competent and ready to settle. So there's no waiting, we don't have to baby them for days. They're just ready to use in experiments immediately. So brooders, for us, this is our Porites is our sort of our lab rat because it's so much easier to work with. It's not typically as interesting in terms of its function and as a reef builder. They're not large corals like the elkhorn and the staghorn and those sorts of things that add a lot of dimension to the reef. But they work really well as sort of lab rats because they are easy to get the larvae from, and then we can take our results from these experiments and apply them to the spawning corals. So some of the stuff that we did first, just as sort of a proof of concept to see if coral larvae are even responding to any kind of bacterial biofilms. And that we took these tiles, these are aragonite tiles, we put them out on the reef. You can see here, they're just attached out on the reef, and left them for either 15 or 21 days to develop a biofilm on the surface. So we wanted to leave them long enough that they would get a good microbial biofilm, but not so long that they would start to have other things settling on them. We didn't want a lot of sponges and tunicates and other things growing on them. So then we did some assays with the Porites astreoides larvae, these brooding larvae. And in fact, they would settle on tiles that had been conditioned on the reef. So you can see here, those tiles that were conditioned for 21 days had about 25% settlement. Those that were conditioned for 15 days had a significantly lower amount of settlement. So they will settle on biofilm tiles, and the age of the biofilm seems to have an influence on the amount of settlement we see with this particular coral. And then you can see here this FSW. This is a filtered seawater control. So this is just like a blank tile with nothing. It was never conditioned, and it's just in filtered seawater. And you'll see these in most of the graphs I show because this just shows that without some sort of cue, these larvae won't settle. They have to have something there to trigger them to settle and metamorphose. So to sort of further figure out if we could really say this was a result of the bacteria that had settled onto the tiles, we treated those tiles, those 21-day-old tiles, we treated those with antibiotics. And you can see this drastic drop off in settlement on tiles that were treated with antibiotics. So it seems like they really are responding to living bacteria within the biofilms on those tiles. And so from there, we wanted to know if it's just the biofilm itself, like the mixture of bacteria on those biofilms, or can that activity be attributed to any individual bacterial strains associated with the biofilms? And so to look at that, we isolated bacterial strains from different species of CCA. And then we set up this assay. And for this assay, we had these ceramic discs, and each disc is coated in a biofilm of an individual bacterial strain. So I isolated a little over 20 bacterial strains, and then I had several controls. And so what you're seeing here are 5 replicates for each strain and each control times, I think, 25 different treatments. So we had about 125 Petri dishes with ceramic discs in there. And then you can see in this picture, these black dots are actually the larvae. So that kind of gives you an idea of size for these little guys. You can see them with the naked eye, but they're pretty small. And so I believe for this experiment, we had 20 larvae in each dish. Anyway, it was a lot of counting. There were a lot of larvae, a lot of dishes, and a lot of counting. A little bit of nervousness for a new postdoc, thinking this may be a shot in the dark because I might not have any of the, fairly limited number of bacterial strains that I was testing. I thought there was a good chance that none of them may be active. But I got really lucky. And in fact, this strain that's designated PSMBA5 was active. So it induced settlement. You can see nothing else really induced settlement across the board. So, but this one strain did in fact induce settlement. I ran the 16S sequencing of this strain, and it belongs to the genus Pseudoalteromonas. One of the interesting things about this strain, so this was a strain that we isolated in Florida, and induces settlement of this brooding coral Porites astreoides. When you run the 16S sequence against the database, it is nearly identical to the 16S sequence of Pseudoalteromonas A3, which was isolated in Australia, and induces settlement of a couple of these spawning corals in Australia. So I think it's really interesting that we have very similar bacteria halfway around the world doing very similar things. And so we wanted to then determine if there was a compound that PS5, Pseudoalteromonas PS5, was producing that was responsible for that settlement induction activity. And so we grew that bacterium up on agar plates. We just grew lawns of the bacteria and then scraped them off, freeze-dried them, and then extracted the compounds from the freeze-dried bacterial cells in organic solvents. And then we just added those extracts to filter sterilized seawater in these 6-well plate assays. So you can kind of see here, these 6-well plates are empty except for seawater and then whatever chemical extracts were added. These that you can see here are actually little pieces of the crustose coral and algae Hydrolithon borgesonii, because we use those as positive controls. So we added just the extracts, we added larvae then to each of these dishes and looked at settlement after 6 and 24 hours. And so this might be a little hard to see sort of the depth of perspective here, but what I want to point out here is that some of these larvae attached, so here they're either attached on the bottom or on the side before they metamorphosed. while we did have some that didn't attach to the dish, so they just floated on the surface and they still metamorphosed. And I wanted to kind of point that out because I'm going to talk about the results in terms of how many were fully attached and settled and how many were metamorphosed in the water column. So this is just a crude extract. So this is basically everything we could extract with the organic solvents that we used. This is probably a mixture of hundreds to thousands of compounds in these extracts. And we tested these with both Acropora palmata, that's the first one here, the elkhorn coral, and Porites astreoides. Now, Acropora is a spawner and Porites is a brooder. So typically we get lower settlement with Acropora than we do Porites in general whenever we do assays with these corals. But what you can see in this figure is that here, this first bar is our extract. So if we include all of the larvae that metamorphosed, we had nearly 100% metamorphosis in response to this compound. We had about 55% total metamorphosis in our positive control, the CCA. But what's really most important to look at is the actual amount of fully settled coral larvae. So these are attached and metamorphosed. So we're most interested in that because if they don't attach first, they're likely not going to survive. So once they've metamorphosed in the water column, if they haven't attached to something before that happens, then they're likely not going to survive past that. But what you can see is that we actually have very comparable amounts of full settlement, attachment, and metamorphosis in our extracts compared to our positive control, the CCA. And then this last control here, this DMSO, this is what we call our solvent control. So anytime you add an extract to seawater, you have to do that in a solvent to be able to add it in. And for these assays, we use the solvent DMSO. And so we always do a control with just DMSO to show that the DMSO itself isn't responsible for whatever inductive activity we're seeing. And so we saw very similar things with Porites astreoides. Of course, like I said, the settlement was much higher. We had about 70% settlement for Porites and then in our extract, and then we had a little over 80 for our CCA, but these numbers are fairly comparable. So that was pretty exciting. So we now have a single bacteria that we have isolated that we know induces settlement, and we can say it produces some sort of compound that we are able to extract and get the same activity from. So that's pretty exciting. But of course, from there, we want to try to understand what that compound is. And so to get to that point, we go through a process called Bioassay-guided fractionation. And this is basically a process by which we take that crude extract that I told you has probably hundreds to thousands of different compounds in it, and we start to break those sort of into sections. So let's say we have 100 compounds and we can break it into 4 groups of 25 compounds and then test those groups and then continue to do that sort of thing until we get down to 1 compound. And so we started this process, this bioassay-guided fractionation, with a technique called column chromatography. So the way that this works, column chromatography separates compounds based on polarity. So you can see the column here. So this column is packed with a material. In this case, I think it's silica gel. And then we add our extract to it, and then we run different solvents through of different polarities. And as those different solvents run through, some compounds are more similar to the solid material that's packed in the column, and some compounds are more similar to the solvent that's going through in terms of polarity. So things that are more similar to the solid material, we call that the stationary phase, they will sort of be attached more tightly to that material, so they will flow through slower. Things that are more like the solvent that's going through in terms of polarity, they will spend more time attached to the solvent, and so they will move through faster. And so we can change the polarity of the solvents as we move them through, and they will pull those compounds through based on their polarity. And so you can kind of see that process here. You can see things are separating out into these bands of different colors. So these would be different colored pigments and things that are coming out at different times. And so we separated the crude extract into 4 parts using this technique, this column chromatography, and then just went again and tested those 4 different fractions with Porites astreoides. Again, I told you this is our lab rat because we can get the most done with Porites in a year. I think I forgot to say that Porites will also release larvae in April, May, and June in Florida typically. So we also have 3 chances to sort of work with this coral in a year as opposed to 1 with most of the spawning corals. So you can see here that of those 4 fractions, the activity is all in this first fraction. So this first fraction is inducing about 80% settlement. And that's, again, very comparable to our positive control, this CCA here. So then we further fractionated that fraction using a technique called high performance liquid chromatography. It works on the exact same principle, except that you're running it through a machine, so it's a little more fine scale. And instead of just sort of looking visually at those bands as they're coming through the column, you're actually using a UV detector. So as the compounds go through the column, they then go out and pass through a UV detector. And every time you see something that has UV activity, you'll see a peak on the UV detector. So this is sort of what our HPLC chromatogram looks like. We tested these 10 different fractions that came out of the HPLC, and all of the activity was in this one fraction, number 4. And then we went on to identify that compound as tetrabromoperol. So that's the compound you're seeing here. So we tested that purified compound first with Porites astreoides. So these are results after 6 hours, which is really fast for these. Typically when we do assays with Porites astreoides, we're looking at settlement after 24 hours. So the fact that this compound induced settlement so quickly is really interesting. So what you can see here, and also this graph is sort of divided into 2 parts. And the reason for that is that we actually did this as 2 separate experiments, but I wanted to show it all together. So this is looking at a range of concentrations of that compound, tetrabromoperol, in nanograms per milliliter. So we tested it from 6.25 nanograms per milliliter all the way up to 500. And what you can see here is, there we go, that we have in our positive controls for these 2 separate experiments, our CCA, again, this is hydrolithon borgesonii, induced 46 and 62% settlement in these 2 assays. And our purified compound is comparable, if not more, inducing comparable, if not more, amounts of settlement for Porites astreoides. One of the other interesting things about this is when we tested at the highest concentration up here at 500 nanograms per milliliter, you can see this large drop off in settlement. And it actually, at this high concentration, was toxic to the larvae. So we saw a lot of the larvae dying at this high concentration. So I guess this would just point out that it's really important. The concentration of a compound and a lot of these signaling molecules is really important. So we did these similar assays with the spawning species. So this is sort of what I was saying, is we like to try to get down to the fine tuning with the Porites and then go out and test like the compound that we've isolated with the spawning species, because we have such a limited ability to get larvae from these spawning species. So we did test this compound with Acropora palmata and saw very similar results. Here we see our positive control had 20% settlement, and the highest amount of settlement we saw with tetrabromoperol was double that at 42% settlement here. And then we did a similar experiment with Orbicella franxi. And again, our positive control, we had about 12% settlement, and at the highest impact, we had triple that with 36% settlement with the isolated compound. And then just sort of as a sanity check, just to kind of double check that this compound isn't somehow just causing these larvae to misfire and settle in some weird unnatural way. We actually followed the settled corals for Acropora palmata for 4 weeks. So we monitored them for 4 weeks after settlement, after they had been exposed to this compound. And there was no difference in survival between those that were exposed to the compound and those that weren't. And you can see here, this is an example of a growing juvenile Acropora palmata, and you can see that it's growing quite normally. It's laid down this nice skeleton. You can see it's budding some new polyps. So here's the main polyp, but then there are new polyps budding off on the side. You can kind of see this one peeking out. And it has taken in zooxanthellae. So this species, Acropora palmata, does not have zooxanthellae as a larva or as a newly settled coral. So you can see these are actually larvae of the Acropora palmata. They're not brown like the Porites are. So these don't take up zooxanthellae till after they're settled and as they start to grow. So the fact that it's budding and it's taking up zooxanthellae, it looks fairly normal. We thought those were all pretty good signs that this compound is not harming these corals in any way. Whoops. And so sort of follow that up, we started working with Danielle Dixon from University of Delaware back in 2014 or 2015, I think. And Danielle does a lot of really interesting work looking at waterborne chemical cues and the attraction or deterrence of larvae to those cues. So in this setup that she has here, what she actually has is a little 2-channel flume. And she has source water here coming from either side. The water is flowing then into the flume, and you'll have 2 different sources of water at any given time. And so we did some of these assays looking at tetrabromoperol in the water to see if the coral larvae would be attracted to the tetrabromoperol in the flume assay. And so in these assays, you can actually put the coral larvae in, and they will swim to one side of the flume or the other. And then you look at how long they spend in one side or the other based on whether they like what's coming from one water source or the other water source, basically. So we did that for Acropora palmata, Acropora cervicornis, and Porites astreoides. And each time you can see that there was a real attraction to water that had tetrabromoperol in it. And this is at a fairly low concentration. This is only 25 nanograms per milliliter of tetrabromoperol. But they are definitely choosing to be on the in the stream of water that contains that compound. So not only does it induce settlement on the surface, it is attracting coral larvae in these flume assays as well. And then we also did some work with Brad Moore, looking at, he's really interested in the biosynthesis of some of these compounds. And so he and his students had been looking at the biosynthetic pathway of this tetrabromoperol and some of the other chemical compounds that are produced along that pathway. And so he sent us a bunch of those different compounds, and they're all across the bottom here at different concentrations. So there's different compounds, and each compound was tested at 3 different concentrations. But basically what you can see is the only compound of this group that had activity in terms of causing settlement was tetrabromoperol. That's in the orange bars here. So these caused significant increases in settlement. This was with Porites astreoides also. And then here's, again, the CCA control. So we see this compound inducing settlement It's acting as an attractant in the water column. It's very specific activity to this compound. Some of these compounds that we tested are very similar. For example, the blue bars here, this is tribromoperol. This only differs from tetrabromoperol by 1 bromine. Basically, there's 3 bromines here and there's 4 bromines here. And that's the only difference between these compounds. So it's a very specific response to this compound, which is very interesting. So yeah, just sort of summarize that. What we're seeing is this compound that's being produced by a bacterium that's associated with CCA. It does induce multiple Caribbean coral species to settle. And I believe that, I personally believe that it's probably fairly universal, because these results that I've shown you are already looking at 2 different reproductive strategies. So we have brooders and spawners responding to it, 3 different genera of corals. I've also tested the crude extracts with just Acropora cervicornis, but I never was able to test the purified compound because of difficulties getting larvae from that coral. I've also tested it with Pseudodiploria strigosa. Unfortunately, Pseudodiploria strigosa larvae settled in response to our solvent control, so I can't make any real hard conclusions about that. But I really think if we continued to test this with other Caribbean species, we would see similar results. So some of the things that I'm really interested in working on going forward are trying to determine if we can isolate other settlement-inducing bacteria. How common are these bacteria on the reef? So obviously we've done all this work in the lab and we've isolated this bacteria and grown it in the lab and it's producing this compound. But what's actually going on the reef? Is it producing the compound on the reef? Are there other bacteria producing this compound on the reef? We know there are other bacteria capable of producing this compound. So I'm really interested in moving forward, trying to look at some of those factors to see how prevalent this compound is, how much it may actually be influencing recruitment of corals to reefs, and that sort of thing. And then how climate change and other environmental stressors affect these bacteria. So we're really, often hear people talk about the effects of stressors and climate change and things like that on macroscopic organisms like, corals or sponges or algae or whatnot. But we are learning more and more all the time how important these bacteria are. So as I said in the title slide, they are small, but they are mighty. And bacteria are really influencing a lot of the health and interactions of these organisms on the reef. And so there's actually relatively little done or little known about how some of these stressors, environmental stressors, are impacting the microbial communities associated with benthic organisms, associated with the benthos itself, those sorts of things. And so we have been doing some work. I've been doing a bit of work with Justin Campbell. I'm sure some of you know Dr. Campbell. He was at the Marine Station with us for a little while, and we did a number of things looking at the influences of climate change and macroalgal contact on larval settlement and microbial communities associated with CCA and that sort of thing. So we have been working on some of that, and I think that's an area that we will continue to explore more in the future. So I just want to thank a number of people. So this work obviously it's very intense to work with these spawning corals especially, and so that can't ever be done without a large team of people helping. And so this is a list, but by no means an exhaustive list of all the people that have helped through the years working on these projects. And of course, the Smithsonian Marine Station, where I'm based, the Caribbean Coral Reef Ecosystem Program, that's based out of Caribou Key in Belize. And then Moat Marine Lab and the Protect Our Reefs grant program that helped fund a lot of this research. And with that, I am happy to answer any questions.
Great, thanks a lot, Jennifer. I'm sure people are clapping out there in internet land. Unfortunately, that's one of the things you miss out on. And
yeah, it's a little weird.
talking of missing out, those pictures of a glass out of Caribou just remind us what we're missing out with.
Oh.
But so we've got one question coming in on chat. People can unmute themselves. We're a relatively small group, or just use the raise your hand. I wonder whether I could just get it started by just asking you the relatively obvious question, I guess, is can we use this knowledge about this chemical to help reef restoration maybe, or sort of increase natural settlement rates on reefs that are recovering?
Yeah, so I'm not so sure about using it quite yet for natural restoration, because there's just so many unknowns as to what it might do besides induce settlement. We know that it can act as an antibiotic against certain bacteria and things like that. So I'd be a little hesitant to just put it out on the reef per se. But we have been discussing with Margaret Miller at Secor using this in some of her restoration efforts.
Right. So I don't know whether Jean has access to the microphone, but just in case, he asked, what are the concentrations of TBP around CCA in nature, and do you have any sense of how this would differ among different species of CCA?
Yeah, so that's something we really don't know at all. So that's part of what I was trying to say in this sort of future research projects, is trying to get an idea about that. These things can be really tricky because they're active at such low concentrations that often when we try to detect them in the environment, our instruments are not able to detect them at the level that the coral larvae are able to detect them. So even if it's there, it could be there in such a low concentration that a coral larvae could sense it, but our instruments cannot. And so that's one of the challenges, and we're working on coming up with some ideas of ways to look at those questions, and then of course trying to get funding to do that. So
as ever.
Yeah.
Hey, anybody else out there? So again, just in case
something popped up, I think.
Joel asks, did you decide what microbes to screen for? Might there be other microbes involved?
Oops, sorry, I'm pushing things. It was pretty random. I mean, it was just things that I could isolate on marine broth agar. So when I first did it, that's why I sort of said it was kind of a shot in the dark. I isolated, I think it was 22 or 23 different bacterial strains, and I just tested the ones that I isolated. So the ones I isolated are probably, they're going to be, the things that are most easily isolated. And definitely it's worthwhile to sort of look at different culture media, different culture conditions, and try to isolate and test more different strains of bacteria. It's just challenging, I think I tried to portray how challenging it is to get the larvae in general to do these experiments, and then the manpower and time to do the assays themselves is very, it takes a lot of time and manpower. So even to test those 22 strains, we had 125 dishes that had to all have, a ceramic dish in it that was coated with a bacteria. Then it you had to count, 20 larvae into each dish. And then you have to count them again after 6 hours or 24 hours, to see how many settled and how many attached. And it just, it can get very overwhelming and out of hand. So it makes it a bit tricky to try to do a lot of different bacteria. So we got kind of lucky that of those 22 or so strains that we did test, we had one that was active. But I think that if we tested more, we probably would find more. And there have been other researchers working with other corals that have found other bacterial strains that induce settlement for other corals as well. So I by no means think this is the only one that induces settlement.
Sure. Okay, any others? I'm seeing some appreciation in the chat, so thanks for that. Okay, well, I'm not seeing anything else at the moment, so I think we're at 3 o'clock. So probably people moving on to their 50th Zoom call of the day. So just once again, thanks a lot for coming online and doing this in slightly unusual circumstances, but it came over great. Thanks a lot, and I'm sure you'd be happy to take any other questions by email, or if people are in Fort Pierce. So on behalf of this group, we thank you and wish you success with this work, and let's hope we can all get back out into the field soon.
Yeah, thank you. Thanks for the invitation.
Okay, thanks, Jennifer.
Take care.